The following is a presentation on modeling human disease and tools for cell and tissue culture. This presentation is by Holly Hattaway, Cell Culture Product Manager, PHC Corporation of North America, representing PHCbi brand laboratory products nationwide.
So today we're going to talk about maximizing your cell yield and some of the factors that go into maintaining an ideal incubator environment. So the business need in North America accounts for the majority share of the growing cell culture market. The market is expected to reach $29.2 billion by 2024 from $16 billion in 2019. Cell therapy is growing and diversifying as science's understanding of disease expands. These are historic times for innovative science and cell expansion. As the need for innovative science grows, the products and therapies grow as well. Companies are culturing cells to produce a list of biological relevant byproducts from therapeutic antibodies and proteins to hormones and clotting factors. Stem cell and regenerative medicine are emerging fields that are using whole cell therapies. Facilities are expanding highly specialized cells and cell lines that will be used in therapies to treat a range of disease and injury. There are many emerging cell therapies that create a need for scaling up. Autologous and allogeneic. Autologous from auto meaning self is a therapy that uses the cells derived from the patient, while allogenic from allo meaning other or different is a therapy that uses cells harvested from a different person or a patient. Once cells are harvested, they are then modified, expanded, and introduced or reintroduced to the patient. And I actually have a visual of the workflow on the next slide. Both types of cell therapies have pros and cons. While autologous therapies can have the benefit of being an exact match for the patient, it can be difficult to expand the ideal amount of cells harvested from that patient for proper therapy. Maintaining homogeneity throughout the therapeutic cell culture can also prove difficult. In allogenic cell therapy, the donor will most likely be healthy, allowing for large amounts of healthy cells to be harvested. The obvious disadvantage here is the heightened risk of the patient having a negative autoimmune response. Regardless of which you choose, once cells are collected, they require a similar workflow prior to administration. The expansion step is a bit different and usually done on a scale that would require an incubator rather than something like a bioreactor. Patient cells must be kept in the best conditions possible in order to stay viable. Most often, large-scale incubation is used to proliferate cell lines. There are two main types of mammalian cell lines, adherent and suspension. The more common cell line, the adherent cell line, grows in a monolayer surface. Cell culture, dishes, flasks, and stacks are the more common ways of culturing adherent lines. Roller bottles require an apparatus that can be stored inside the incubator chamber. This can be managed through shelf configuration and reinforcement. Suspension cell lines require an apparatus that create repetitive movement in order to keep cells healthy and bathed in proper nutrients. Agitation and weight from these apparatuses can be also managed through shelf configuration and reinforcement. There are many ways to scale up. And one of the most common ways to scale up starts with T-culture flasks, which provide a slightly larger surface area than common culture dishes. As scale-up continues, larger cell stacks can provide increased surface area in a fairly small footprint. Depending on the type of cell and application, scaling up can mean increased incubator space or large-scale bioreactors. In most adherent cell culture scale-up, especially small to mid-size scale-up, facilities require an increased amount of incubator space. Often large capacity incubators are the solution. An incubator designed to properly mimic the conditions of a living mammalian organism. So we've talked a bit about the amazing scientific applications that are relevant to cell culture scale-up. And now let's talk a little bit about incubation, large-scale incubation, and what features need to be considered and stepped up when you're scaling up your cell culture. Cells are always susceptible to contamination, stress, and death. In mimicking the proper mammalian environment, an incubator must provide a stable, uniform, warm, and humid environment while mitigating contamination that these conditions attract. It's important that large incubators provide superior insulation while also providing a clear view to minimize door openings. A double-panel gas-insulated door provides the added insulation needed to protect the cell culture environment while also providing a clear view. For added control, a 5-inner door option allows for isolation between the solid shelving. It's also great for frequent door openings. When considering the best door options, it's also important to consider a blackout option for large incubator doors if cell lines are light sensitive. Having a well-designed incubator door with the right options in place is a big part of ensuring a stable, uniform environment for your cell cultures. A uniform, stable incubator environment can be achieved through a combination of construction and design. Perforated side plenum walls create a positive, even, unblockable airflow, while horizontal airflow allows for full use of solid shelves, which allows for added insulation benefits that I'll discuss later. And upon door openings, airflow is halted. This prevents against an excess of air exchange between the outer and the inner environments, and this guarding against chamber variations and contamination entry. It's important that the incubator environment maintain a uniform, even airflow. Variations in temperature, humidity, or CO2 can create inconsistencies in cell homogeneity, quality, and viability. Healthy cells rely on proper amounts of atmospheric CO2 to be readily available. Cell media depends on the delicate balance of dissolved carbon dioxide and bicarbonate to maintain proper pH levels for healthy cell growth. Changes in the atmospheric CO2 can alter the pH of the medium and cause detrimental changes to the cell culture, including death. It's important that every cubic inch of the incubator chamber is saturated with proper levels of carbon dioxide, most often 5%, to ensure CO2 is at the proper levels and it stays there. An automatic tank switcher can also ensure that there is plenty of CO2 readily available for use. Humidity must be consistent throughout the entire incubation chamber in order to maintain the proper osmotic balance a cell needs to thrive. Humidity levels can be set to 80% humidity for normal humidity conditions. The humidity levels can also be set to a 90% relative humidity to facilitate an elevated humid environment. While important to cell function, humidity attracts unwanted contaminants. It's important that all humidity sources are kept clean and mitigate possible contamination. It's also important to have humidity control for changes in humidification requirements brought on by large-scale apparatuses. Because humidity is so important to maintaining healthy cells, it is important to always make sure that the humidity source is amply supplied with water. Increased incubator capacity creates an increase in humidity demand. A large capacity incubator should be designed to maintain water levels above 5 liters at all times. It may be beneficial to consider an optional additional water supply to aid in humidity demand. Over longer periods away from the lab, an additional water supply can help ensure that the humidity source doesn't dry out. It's also important to make sure that the water is supplied gradually to the humidity source to avoid variations in temperatures. Other conditions, like a very dry climate or dry ambient conditions, can create a need for increased humidity as well. Adjusting the levels for the humidity source can be done easily from the front of your incubator. It's important to be able to easily control the parameters of your incubator. Temperature, humidity, and CO2 can be controlled all in one place from the front of the incubator. CO2 and temperature display can be seen from across the lab, and standard alarms for temperature, chamber, CO2 tank supply, and water levels help safeguard optimum conditions in the incubator chamber. As well, a standard key lock for user security helps safeguard incubator set parameters. As incubator size increases, heat must be controlled and monitored from several different heat sources. A main heater, bottom heater, flange heater, door frame, and door heater all combine to maintain and recover optimal incubation temperatures. After someone has had the door open for 30 seconds, all heat sources act to restore the internal environment's ideal temperature in minutes. A separate heat source for the humidification pan is in place to control the different levels of humidity. Historically, the most common type of CO2 sensors were thermal conductivity sensors. These sensors measure the resistance of CO2 using the chamber's ambient air as a reference. Fluctuations in temperature and humidity can affect a TC sensor's ability to read the CO2 in the chamber. Infrared sensors measure the CO2 gas in the air directly. This type of sensor is unaffected by changes in incubator temperature or relative humidity. In order to accurately maintain desired CO2 levels, an infrared sensor measures the atmospheric CO2 in the gas directly. And as we have mentioned before, door openings can cause big changes in an incubator's internal environment. This allows the right amount of CO2 to be quickly delivered to the incubator chamber, restoring the proper CO2 levels without any overshoot. An IR sensor enables the incubator to rapidly recover that CO2 level after the door opens. When the incubator door is open, the CO2 density inside the chamber can drop around 3%. In 2 minutes, the IR sensor brings CO2 levels back up to the ideal atmospheric conditions. And again, door openings can pose many threats to the internal cell incubator environment. As we all know, contamination is the biggest threat to any cell incubator. Mitigating contamination is imperative to successful cell culture. The most common type of incubator, historically, is made with a stainless steel interior. This is because the stainless steel does a good job at withstanding the warm, humid conditions of a cell incubator. Stainless steel's strength and resistance to corrosion make it a great candidate for a variety of scientific applications. Some companies have decided to use copper instead of stainless steel to create a premium option for cell culturists who are concerned with contamination. Metallic copper surfaces kill bacteria, yeasts, and viruses. Having a completely copper interior can be beneficial in that it mitigates contamination. The major issue with these interiors is that they do easily corrode and become extremely difficult to clean and keep in good conditions. A third option was created to exploit the benefits of both types of metal. While overcoming the issues associated with corrosion, a stainless steel copper alloy provides both the antimicrobial benefits of copper, as well as the strength and the corrosion resistance of stainless steel. As a result, an incubator with a stainless steel copper alloy interior combines the germicidal benefits of copper with the corrosion resistance of stainless steel. Bacteria, yeasts, and viruses are rapidly killed on metallic copper surfaces, and the term contact killing has been coined for the process. Copper is an essential nutrient that is taken up by contaminants. After taking up large amounts of copper ions, microbial membrane walls rupture, the internal osmotic pressure is disrupted, oxidative stress generates hydrogen peroxide, and the microbe is destroyed. Beyond passively mitigating contamination, an incubator must actively remove contaminants from the environment. Most of you are familiar with closing a hood and flipping on the UV lamp to bring the BSC workspace back to sterile conditions. Ultraviolet light has been commonly used in the lab to sterilize metallic surfaces and instruments that cannot be autoclaved. Light emitting 253.7 nanometers in wavelength destroys bacteria, molds, spores, viruses, yeasts, and fungi. Hidden behind a plenum wall, the UV penetrates the water and air that will flow through the internal chamber of the incubator. Again, opening the door allows contaminants attracted to the warm, humid environments to get into the incubator chamber. Once the incubator door is closed, the UV emits light that kills newly introduced contaminants from anywhere from 5 minutes to, or depending on your user preference, a half hour. Unlike the traditional HEPA filter, UV light destroys 99.999% of all contaminants, instead of trapping them inside the incubator space. Heavier configurations, usually orbitals and shakers, can create a strain on the incubator shelves. This can be overcome by configuring reinforced shelving for added stability. We have two types of configurations in place that are best suited for different applications. The 3 reinforced shelf option available was configured with cell shakers and orbitals in mind due to the weight and space demands of this application. The 4 reinforced shelf option was configured with heavier cell stacks in mind and accounts for weight and spacing for this application's needs. Individual and custom configurations can be fit to specific needs. To maximize space, roller bottle apparatuses can be used in a large-scale incubator. A large apparatus can create extra heat and affect the incubator's relative humidity. Microprocessor settings can be controlled to offset heat created by an apparatus or apparatuses. An optional mount designed to securely install the roller bottle apparatus can be used. To maximize space, roller bottle apparatuses can be used in a large-scale incubator. A large apparatus can create extra heat and affect the incubator's relative humidity. Microprocessor settings can be controlled to offset heat created by an apparatus or apparatuses. An optional mount designed to securely install the roller bottle apparatus can be used. Having an unblockable horizontal laminar airflow is essential when adding apparatuses to an incubator. Remote monitoring of cell culture is important when scaling up. Cloud-based remote monitoring, Lab Alert, lets you monitor humidity levels, CO2 concentration, and temperature from your phone, tablet, or personal computer. Easily visualize the parameters through graphical format, and all data is stored and backed up in your encrypted archives. Our MCO 80ICL cell production incubator is the perfect incubator for a lab that is looking to scale up their cell culture, providing a state-of-the-art environment for repeatable cell growth and yield, while mitigating the external threats that contribute to cell contamination, stress, and even death. The IncuSafe copper-infused alloy passively mitigates contamination while remaining corrosion resistant and easy to clean. The ultraviolet lamp actively decontaminates the humidity source safely behind a plenum wall. Infrared sensors measures atmospheric CO2 directly for uniform, precise CO2 control even after door openings. Monitor and control parameters with a user-friendly microprocessor controller. If you are planning on using a heavier apparatus or apparatuses in your incubation, reinforced shelving can support your configuration. And a double-pane gas-insulated door guards against temperature, humidity, and CO2 fluctuations while maintaining a clear view of your cells. The cell production incubator was designed with side-perforated walls and horizontal laminar airflow to ensure a stable, uniform environment throughout the chamber and a quick recovery after door openings. An optional external 20-liter water supply was designed with the added humidity needs in mind. And that is the presentation. Thank you.
You've been listening to a presentation on modeling human disease and tools for cell and tissue culture by Holly Hattaway, Cell Culture Product Manager, PHC Corporation of North America.